broadcast. We'll broadcast in 15 seconds. So
Yes.
let me start the session.
Can I share my screen?
Yeah, you're welcome. Okay, so thank you for joining us. Thank you for joining us in this continued discovery track. And it is now my pleasure to welcome Pankaj Daga, who is a principal scientist at Simulations Plus, focused on data science. And Pankaj will be talking to us about modeling CYP sites of metabolism. So
Can you
uh,
see my screen now?
me maybe before we start, let me launch a quick poll to see how this topic is of interest to our audience. So I'm going to launch the poll for 30 seconds and let it run. So please let us know if this topic of modeling CYP sites of metabolism is very important to you, somewhat important, or not important, and we hope that it is. Okay, I will retrieve the poll now. Thank you for submitting your information. And it looks like we have about 75% of our audience who thinks it's really important to them. So they certainly look forward to listening to you, Pankaj. So let's go ahead. The floor is yours.
Thank you so much, everyone, for attending this discovery track, and welcome to the second second talk of this session. I have modified the title of this presentation a little bit to make it suitable for the content. So instead of just talking about CYP metabolism, I'm going to talk about how we are going to predict the CYP reactivity and sites of metabolism in ADMET Predictor. So as we all know, the drug discovery is very time consuming and expensive, and it is labor intensive. What makes it worse is the failure of drugs during the clinical trials. Up to 25% of the compounds are withdrawn from the market or from the clinical trials due to metabolic, pharmacokinetic, or toxic problems. And this metabolic liability can lead to various complicated issues, which can be something like drug-drug interaction or safety hazards of the metabolites. So having knowledge of metabolism and having knowledge of metabolic uh, products of the small molecules during the early drug discovery can be very useful. So if we can predict what parts of the molecule metabolic reactions, or that or in that way we can find out which are the portions or which section of the molecule can be modulated or modified to update the safety and efficacy or to modify its metabolism. Furthermore, if we know which enzymes or which CYP isoforms are responsible for metabolizing the newly designed molecules, we can restrict those drug-drug interactions, or we can predict those drug-drug interactions ahead of time, and then thus we can modify the drug's applicability. And furthermore, the most important part is if we know if the metabolized products, if the metabolite of the new compound is toxic or non-toxic. That way, if that compound is really important, we can update, we can focus on administering it carefully, or we will know that this compound has low therapeutic index. So this makes the drug metabolism very important during our early drug discovery. CYP 450, or we call cytochrome 450s, or we call it CYPs. These are the primary macromolecular compounds which are responsible for almost 80% of the 80% of the drug metabolism in the market. Those are available in the market. These are superfamilies of the enzymes that contain the heme as cofactor, five major SIPs that have been uh, reported to be very important and responsible for xenobiotic or drug metabolism: 1A2, 2C9, 2C19, 2D6, and 3A4. I have highlighted 3A4 intentionally here to mention that the 3A4 is responsible for almost 70% of the drugs which are metabolized by overall SEP class of enzymes. There are four other SIPs which are also studied by various groups, but their contribution in drug metabolism is very minimal. And these four SIPs include 2A6, 2B6, 2C8, and 2E1. And one thing which I like to mention in here is some of the drugs are metabolized by multiple CYP isoforms, but there are some drugs or some small molecules which are metabolized only by one CYP isoform. And our goal is to look at all these CYPs which can be look at these CYPs which are responsible for metabolism of these small molecules. There are predictive CYP models. I'm not going to detail all the ones which are available in the market, but the goal of predicting these CYP sites of metabolism is very straightforward. If we want to determine experimentally what are the sites of metabolism or what are the metabolites of the new drug, it is a very expensive and time consuming task. But in silico predictions of sites of metabolism and metabolites, it helps to reduce the cost and the time in the drug discovery process. Generally, or roughly, we can divide these SOM predictions. I call SOM, that is sites of metabolism and metabolite predictions, in three classes. One is rule based class, structure based, and ligand based. Rule based determines the possible metabolites of the small molecules and predicts the SOMs using the dictionary of biotransformation rules. The major hurdle over here is this list is compiled by human experts. So it is more or less human dependent list. When we look at the structure based approach, the structure based approach and the objective here is to study the protein ligand interactions, and a lot of CYP crystal structures have been solved in the literature which can be used to study these interactions and possible sites of metabolism. But the issue over here is it requires high quality crystal structures, it requires higher computational cost, and very time consuming. And not to mention, there are other factors which are contributing to it, which includes docking, scoring functions, and flexibility of CYP isoforms. So the major concern with structure based is the time consumption and the requirements of high quality structures. The ligand based approaches, these are the ones which mostly use the machine learning methods and atom level descriptors with large amount of high quality data available from the literature and reliable set of descriptors which are available nowadays in the literature. The ligand based methods can be comparable with the structure based outcome. Of course, these methods rely heavily on available experimental data as of sites of metabolism as well as metabolites. Without further going into more details and spending more time on SIP introductory session, I will like to reach out to you. I would like to mention what are the SIP ligand interactions that we have available in ADMET Predictor. There are five different groups of properties that can be predicted using ADMET Predictor. First one is selectivity, which suggests which isoform is active against the ligand. That means if the compound is being metabolized by the CYPs, which isoform is responsible for substrate for this metabolism, or if the compound is inhibiting the proteins, inhibiting the CYPs, which isoform it is going to inhibit. So that selectivity is followed by the reactivity. When I say reactivity determines if the compound is metabolized by CYP, that means binds to the CYP if it is a substrate of the CYP, or if inhibits the CYP, or if it is inhibitor or non-inhibitor. There is third kind of activity as well, CYP inducer, induction or non-induction. Uh, we are not going into those details right now. Third group of properties which we predict in ADMET Predictor is regioselectivity. Some people also call it sites of metabolism. It this this group of property determines which specific part is going to be metabolized. What are the sites of metabolism? If we know sites of metabolism ahead of time, we can shut off the metabolism by proper substitution in that compound, like CH can be substituted with CF, and further different substitutions can be taken into account. Next group of properties is metabolites. ADMET Predictor predicts likely metabolites of the small molecules using information obtained by these regioselectivity models, as well as a dictionary of biotransformation rules, which are included along with the ADMET Predictor. Knowledge of these metabolites can be very useful to determine if the compound might lead to toxic or non-toxic metabolites. And last not but the least is the kinetics. These are quantitative properties like Michaelis Menten equation, Km value, or Vmax, or clearance. And these properties are used um, mostly during our drug-drug interaction predictions. The primary focus of this presentation is only selectivity, reactivity, and regioselectivity or sites of metabolism. So let's focus on only selectivity and reactivity combined. And as I mentioned, the focus is only on the substrate or non-substrate models at this stage. CYP substrate, there are 9 CYP substrate classification models present in ADMET Predictor, which I have already mentioned in the beginning slide. There are 5 major and 4 minor minor CYP isoforms which we have models for. The major concern or very important aspect while building the models is data collection and data curation. It is absolutely necessary to use high quality and correct data sets to build the good and predictive models. So our models, uh, to build these models, we have collected the data from various sources, and first and primary source was various compiled data sets from the literature, which are made available from various research groups like Sheridan and Zaretsky group. We also used commercial and academic databases, and we scanned through hundreds and hundreds of FDA review submission files, which are available from our FDA site, to find out the CYP class, CYP metabolism for the approved drugs. And of course, we have scanned through the literature as well. While collecting the data, we made sure that the compounds were included even if the specific metabolite is not reported. For example, if we look at this table, we can see that in some cases CYPs are responsible, which leads to a specific metabolites. We have extracted the chemical structure, but in some cases we have seen that CYP, when the compound is treated with the CYP, it just authors report its disappearance, but not a specific metabolite. Even then, this compound is a substrate of reported CYPs in that case. And that's how our substrate database was compiled. Once we have compiled our substrate dataset, next step was simple but very expensive data curation. We make sure that we follow a data curation to remove all the possible errors from the datasets, and it it includes correcting the chemical structures or correcting the sites of metabolism. The table here, we can see that these are the some of the compounds which we which we acquired from the compiled datasets from Zaretsky and his research group. This dataset is widely used by a lot of researchers who have been following building sites of metabolism models. When we started looking at this data, we found that there were a lot of errors in the collected in this structure structures as well. For example, we can see that the sec-butyl group is missing from sec-butyl group was the difference of sec-butyl group or addition or removal of the halide groups. So these structural corrections were necessary before we started building the models. And we also made sure that when we compiled the data from various sources, we had to make sure removing identical duplicates. If one data set reports some extra sites of metabolism, we had to merge the data sets. But finally, when we compiled our final data set, we can see that we have almost 3,000 compounds in our substrate non-substrate data set. We had substrate models in our ADMET predictor in the past, but these models are over 50% larger data sets compared to our previous simulations plus data set, which were used to build the SIP substrate models. And if we just want to have a look at the composition of each data set, we can see that almost all these SIP isoforms have non-substrates as majority and substrate compounds as the minor class. Of course, as I mentioned in the beginning, 3A4 is the major contributor towards the metabolism, the SIP metabolism, and this is what we saw in our data set as well. We have large number of substrates compared to the non-substrate compounds available in 3A4 dataset. Now we have the final dataset. Next step was simple model building. Our model building approach includes various steps like selecting the test set, and that test set selection is we have 4 or 5 different approaches to select the test set. But the most important part, which I like to focus, is we have around 300 or more than 300 molecular descriptors available in ADMET Predictor. We use these descriptors and artificial neural network method to build the model, wherein it is we have one hidden neuron finally leading to an output. So this artificial neural network has been shown to be a very promising model building method. However, it has been reported in the literature as well, and we have shown it in the past that instead of relying only on one model, it is better to build multiple models or ensemble of model. So in ADMET predictor, in ADMET model, excuse me, we build around 165 models, and 33 models are selected, which are the best ones. And finally, we determine what is the final performance of these models, which is reported as various performance metrics. In this case, we are looking at accuracy of our classification models, and we can see almost all these SIP substrate models are 75% or more, have 75% or more accuracy. And just to give you an overview how these outputs are displayed in ADMET Predictor, we have all these 9 substrate models, and we also depict it as a star plot. And as I mentioned, these models are all ensemble models, so we also provide a confidence estimate in our predictions, which is calculated using beta binomial distribution. And beta binomial distribution is already being published in the literature, so if anybody is interested, I can provide those references, or you can pull out those references from Simulations Plus website. Once we have the substrate model, next task was looking at the regioselectivity models, or we call it sites of metabolism models. Again, quickly going through the process, we have 9 sites of metabolism models, and it includes around 1400 compounds, over 1400 unique compounds, and 5500 or more unique metabolite sites. When I mention unique compound and unique metabolite sites, this is the demonstration I like to show you. This is a nevirapine compound. The overall nevirapine compound, contains non-hydrogen 20, excuse me, 20 heavy atoms. There are few atoms which we do not include in our study. These are the atoms which are, we call them excluded atoms. These are the ones which have no potential sites, which are not the potential sites of metabolism, and these are non-hydrogen, sorry, carbons containing no bonded hydrogens. For example, these ones, they are carbonyl oxygens or halogens, phosphorus, sulfonyl sulfurs, or quaternary nitrogens. These are the ones which we do not include in our study. So we call them excluded atoms. So that leaves us around 13 atoms which are potential sites in the entire molecule. So we have 13 potential sites, but it has been reported that nevirapine is metabolized at 4 different sites. So altogether, we are talking them as these are the 4 positive sites, and that leaves us 9 negative sites. So this is how we find out our unique metabolite sites, as well as what are the negatives and positives in our classification model. So that way, if we see all these different data sets we have for different isoforms, we can see that we have more negatives and less number of positives. And when you say this 5500 unique metabolite sites, I'm talking only about the positives throughout all 9 isoforms. And another important aspect I want to mention is this number in parentheses. It represents the total number of substrates we had used in our SIP substrate model. And if you recall my previous discussion, there were few compounds which are reported to be just disappearance of the compound while treatment with this enzyme. Those compounds are not included in this data set. Only whenever there is a specific metabolite reported, only those compounds are included for the sites of metabolism. And this gives us overall 45,000 compound 45,000 atoms over negatives and positives together, giving us a very large data set to build our sites of metabolism model. Um, Similar approach, but more complicated approach has been used to build these models. This includes various, when we are building the sites of metabolism model, we add the atomic descriptors as well. And these descriptors are, some of these descriptors are calculated our, uh, by using quantum chemical calculations and running it through our machine learning methods. We had carried out around um, we had carried out optimization of around 1,000 different small molecules, drug-like molecules, using quantum chemical calculations, calculated the partial charges, and that's how actually these reactivity descriptors or Huckel charge methods, Huckel charge models, where we have these descriptors, and these descriptors are used in our sites of metabolism models. Finally, if we just again following through the entire process of ensemble models and getting the final accuracy, we can see that almost all these models predict over 80% accuracy in predicting correct site of metabolism in our in the small molecules. How do we measure the performance of these sites of metabolism? There is, apart from accuracy and sensitivity specificity, which is a conventional scoring metric in um, classification models, there is widely used performance metric. They call it top 2 or top 3 performance metric. Here, what we do is each atom is assigned a score. So when you see, we have a atomic property descriptor, atomic property display in ADMET Predictor, which can give you the scores of metabolite, scores of our SOM models. Each atom is scored by these SOM models, and then all the atoms are ranked. Based on the rank, if at least one of the sites of metabolism is discovered in top 3 positions, then the molecule is marked as correctly positive. So in this case, we know that these two are the CYP2C8 sites, and these are the predictions of 2C8. So if we look at if we rank these atoms, we can see that this is the top ranked position, and this is second ranked atom. Both these atoms are correct, are within top three positions of the scores of the ranks. That means this compound is correctly predicted by 2C8. And if we, this is the top two metric evaluate top two metric performance reported by Zaretsky in 2013. And if we see that the overall Xenocyte was considered to be the best in all these nine models, around 85, average 85 to 87% accuracy or accurate model. And if we look at the performance of ADMET Predictor, current ADMET Predictor, we can see that our models are performing equally or even in some cases even better than the models which were reported in the past. This is just to show them next head to head each other. We can see that both these models, in some cases, it is not performing really good, but we have to keep in mind that the data set, what we have used for ADMET Predictor, is over 50% larger compared to the data set which was used in their model building exercise. I have very few minutes, just 2 minutes left. Um, just want to show you there are these are the five different groups of models which I mentioned in the beginning, and all these models are substrate dependent. So once we have a substrate classification, we also predict sites of metabolism. This is, I'm just showing only one suite of 3 or 4, but we have 5 models for each of these, our 9 uh, CYP isoforms. And just to show you that these are the case, I've recently performed, just predicted the metabolism of ebrocitinib, which was recently approved by FDA last month. And this was scanned through their submission review PDF file, and we can see that all the primary metabolites of this compound, which are 5 metabolites, they are predicted correctly using our new models. I think just reaching the conclusion, we have built our new models, expanded the chemical applicability of our CYP models by building new substrate and regioselectivity models. And these models can be used or applied to wide and broad range of molecules. And the most important part is when we combine these SOM models with predefined set of biotransformation rules, we can predict the possible metabolites correctly for these small molecules.
Right.
Am I over time?
Yep. Just yep. It's great, Pankaj. Yeah. Thank you. Thank you.
That gives you time for your.
Yeah. I will just run the last poll.
Yes,
Yeah,
please.
we're really getting pretty tight on time, but thank you. It's very, very interesting stuff. I will launch the last poll. If you could let us know whether, based on what you saw, how likely you are to deploy ADMET Predictor for the prediction of sites of metabolism. And I will leave the poll up for, just like the other ones, about 30 seconds or so. And after that, I will close the session, and we look forward to seeing you in the rest of the discovery track. So with about 45 seconds to spare or so, I will end the poll and retrieve the results. It looks like people really like what they saw. Thank you, Pankaj. Great contribution and meaningful validation work. And again, I look forward to seeing you in the next talk. Thank you.
Thank you. Thank you.